Next speaker is Michael Schwarzschild from Mass General. He will talk about their current clinical research trial.
Give you an update of neuroprotectants nearing the finish line with a focus on how the kind of under the surface, thank you, under the surface, oh great. Research like is being done under Gia's guidance here at the Udall Center is leading to promising therapeutics. And referencing the framework that Michael Byrne mentioned, I think I'll be trying to convey, hopefully easily, that while there are huge challenges in trying to get to disease-modifying therapy to slow the progression of the disease, ultimately to prevent it, and our track record, as I'll show you, is not inspiring. It's amazing the pace at which that research and our understanding is increasing, and it does give a great deal of hope. And I'm, as Gia mentioned, based elsewhere at Partners at Mass General, our research institute is on the Navy Yard campus where Old Iron Science is, and you can take the Zakim Bridge to go across. We've used this for years, not only to get across, but also as a metaphor for the translational biology from labs like Gia's to the therapies that I'll give you a highlight for. So I first wanted to give you some broad context of that pace that I mentioned. And this is just on the heels of our 200th anniversary of the milestone of the first description of Sir James Parkinson's observation of cases that gave the name to the disease back in 1817. This sort of schematized timeline here highlights the pace at which major advances have taken place. And particularly in the last 20 years, taken us to, just up to this year, I won't go through each of those. Each of those dots is an inspiring advance in our understanding, but I'll just give you a little bit of flavor at the less dense part of the timeline. So there was the observations really just in 6 people back then in England, and then it took 100 years later before we even had an idea of where in the brain that Jerry was highlighting the circuitry is and the pharmacology and the wiring that underlies the movement difficulty. And so on the left, of course, you all out there are the experts in what Parkinson's is clinically. After that was described, as I said, 100 years later is when we learned that in this dark substance, the substantia nigra in the top of the brainstem there, cut away from someone who passed away without the disease. That's why it's black, and you can see that black stripe there, compared to someone who passed away with the disease, where it's blanched because that person lost those pigmented, dark pigment containing brain cells. And so that advance here put into a little bit more of an anatomical context that you might recognize from Gerald's talk. You can see in the pulled away part of the top of the brainstem, here's those stripes of black substance that house the cell bodies of these dopamine producing brain cells that project their wiring into these deep structures that you just got a tour of, where at the nerve terminals, the release of dopamine, that chemical signal that comes out of these cells and gets sprinkled onto the circuit, facilitates normal movement, such that with the degeneration of these dopamine-producing brain cells, that facility of movement is lost as you lose some of that innervation. So that was 100 years ago. Again, just to highlight, then I'll drop off because it's just the highway's too dense here. It took another 50 years or so before a huge step in our understanding and the therapeutic potential of some of this research began to be realized with the discovery that dopamine is that chemical signal that gets released. That took a long time after learning what the anatomical structures were. And not too long led to levodopa, still the gold standard of pharmacological treatment, now matched in some ways and in some patients with electrical therapy. And then sort of reflecting a framework that's also useful, sort of environments and genetic influences into the cause of the disease. It was 20, 30 years ago, 40 years ago, it adds up that we began to get clues about the non-genetic environmental nature and a rash of intoxications that led to Parkinson's in California. It's an interesting story in and of itself that highlighted that things in the environment can have an influence. And then a decade or two later, we started in the genetic age of Parkinson's, where multiple genes, a couple that we'll hear a little bit more about, were discovered in a few instances, even if not that as commonly, can actually cause the disease. In most instances, as we learn more genetics, can have an influence, just like presumably the environmental factors, and I'll schematize that too. We learn more about some of the non-genetic influences and from twin studies, for example, that highlighted that even if you have an identical twin, that doesn't increase that twin's likelihood of getting the disease that much more than if you have a non-identical twin, suggesting that genetics aren't, for a lot of people, and for many on average will know that a family member doesn't have Parkinson's is typical in Parkinson's. Parkinson's disease doesn't, for most people, run strong in the family. Again, without getting too detailed, we're more than a decade back here. We began having insights connecting some of the genetics and environmental factors and frameworks in thinking about how the anatomy relates, with the understanding that one genetic clue that David is going to highlight for us, synuclein, is something that may move through the nervous system and gives it, and has, as we'll hear, I'm sure, given us a greater understanding of how these molecules that are implicated may be causing the disease. And then I'll spare you the rapid fire findings that really have led us to this point, where now switching over to the sort of the clinical prospects with this pace and underpinning have brought us through a number of trials, but with a horizon that is particularly promising. So here, switching now to what might look more discouraging at first, and in fact is the reality that when we get to disease-modifying therapies, and it's important to understand the distinction between the tremendous progress starting half-decade ago with levodopa and amongst neurodegenerative diseases actually having the enviable position where medications and interventions can have a big but only partial impact in the disease. We still don't have anything that's been established to slow the underlying process, the loss of those dopamine-producing brain cells. I won't dwell on the past unduly here, but we've learned a lot along the way. And I'll just highlight in this cradle-to-grave metaphor from a colleague, Alberto Espe, that there are a lot of things in various stages of development or infancy or maturation. And in some sense, I've tried a little bit there to highlight that at this point, as I understand it, there's really only two studies that are taking clues at a late stage or so-called phase 3 development to try to really answer the question as best as we can ask it, are we slowing down the disease? And one is isradipine, and I'm going to focus on, for most of the example part of this brief presentation, on one that I'm helping run called inosine as a medication, and I'll tell you the background for that. There are a number of other candidates at a slightly earlier stage, and I just want to point out the bottom half that you'll be hearing more about from David and Peter, which is sort of an inflection point in my mind, where we're going from some of the best clues that have come from a much better understanding. I think these two examples here that are in the pipeline at a later stage, while any study is a long shot, any target is a long shot, are much better than the evidence, and the design of the trials is much more compelling than it was at the stage of our earlier failures. Still, it's a long shot based on a lot of convergent data, and I'll give you that one example. But some of the greatest excitement is down in this part of the nursery here, which is highlighting how we're taking some of those genetic insights where we know the causality of a few genes, at least in some cases, and gets to this precision medicine concept that you'll hear about, but where we're on terra firma for the biology and what we know is causing trouble, and the interventions, the disease-modifying therapy candidates, are are more rationally selected and are just on the verge of a sort of an explosion of therapeutic potential. So with that, I'm going to just return briefly to this framework that's understandable for all of us, which is these nature versus nurture, genes versus environment. It's not black and white. Of course, there are examples which are close to that. Huntington's disease, sickle cell anemia. If you have the gene, you have a very high likelihood in your life from suffering from that condition. And on the far end, there are things that are exposures, too much sun, too much poison, and almost anybody will get the condition. But in Parkinson's, it really is a mixture of these factors. Of course, there's a temporal association. The genetic influences, whether they're strong in a family or just part of a mixture that increases one's risk, they happen early, and so far, they're harder to manipulate, although some of the most exciting therapies are pushing back into that part of the pathophysiology for the first time to be able to make manipulations. And we're not quite there in Parkinson's, but that's one of the next horizons coming up. This is, I like the sequencing this. I'm a little averse, even though I'm showing it, to the overstating the drama, the bloodiness of the suggestion that after you have a predisposition that a trigger gets pulled. But the idea that something out there, things that we're still trying to explore through epidemiology and biomarker work that might be telling us what was taking that predisposition and pushing some people over into development of Parkinson's, and more importantly, what's keeping other people from getting it, and picking up on those clues and figuring out which of those clues, particularly the factors, genetic or environmental, that might be preventing the disease, and ultimately whether that's a drug or treatment is what we're working towards. So one further sort of framework here to just help you see how people think of the causes or the etiology, and then the pathophysiology or the mechanism by which these brain cells degenerate, giving rise to this neurodegenerative condition of Parkinson's. And again, it's genes and environment. On the genetic side, it's actually a bit dizzying to organize things. Some of the genes have been called PARK1 goes well beyond 13 now. And in a few of those instances, generally rare, getting the genes gives you one a high likelihood of getting the disease. And then there's other genetic factors which are really part of this continuum of lesser and lesser, but still important influences. And we'll hear some examples of intermediate genetic factors that clearly increase the risk that we can now try to target. And that's on the genetic side, and it's really very similar for the environmental side, although we really know much less on that. There are a couple of instances where effectively environmental factors can be causal. I mentioned the epidemiology of a series of drug addiction cases where an emergency room doctor observed that people who were coming down with Parkinson's at a young age, and it was found that they had a bad batch of a heroin analog that contained something called MPTP that turned out to be a great clue to the disease. Certainly was an example of how something in the environment could cause Parkinson's, but that's really a rarity. There's a presumption that there are many environmental risk factors. The twin studies I mentioned highlight that, and I'll just touch on some of the epidemiology, but it's really a mixture of that, of the causes, and then what plays out in the brain cells to cause the degeneration and dysfunction of these brain cells. And there are a lot of ideas. Often they're interrelated. The genetic clues point to a number of these. We're not going to go through all these. I'm just going to highlight one particular example that's sort of tractable: oxidative damage, which has been a theory that's been around for decades and has been largely substantiated by the more recent genetic clues, and put it a little bit into a metaphor as well as to the anatomical framework, back to that upper brainstem section with the substantia nigra, and then it's the loss of those pigmented neurons there, as we already highlighted. It does make sense that in some sense, a sort of a cellular rust with oxygen and metals mixing in can lead to the degeneration and loss there, and more recognizable, depending on what vintage you're from, you know that if you don't take care of a metal machine, you can lose its function as well. And just as we have methods that are worked out old-fashioned ways to prevent that, a lot of research is really focused on trying to figure out what it is, particularly for this mechanism, what's the Rust-Oleum there. And so this is just one candidate that's reached the threshold of these late-stage clinical trials that we and colleagues have been working on, where one possible candidate is a natural antioxidant in us called urate. And I'm going to quickly run through, and I want to be cognizant of time, because this is really just an illustration. If you got a sense already of the pace at which work is going on and a couple of examples on the genetics that we'll hear in a few minutes, you'll have gotten the take-home here. But it's an interesting story, and it's an example of how one can go from the research at multiple levels in the labs and the clinics to get to a candidate and then to begin testing it. And this particular example, again with our metaphor from the Zakim Bridge, starts interestingly in evolutionary genetics of gorillas and humans and our ancestors that led us very locally, as it turns out, through some wonderful collaborations here at the Harvard School of Public Health to studying epidemiology of Parkinson's and how this clue jumped out. One of the other take-homes here that you've probably appreciated is that the progress that we've made and the prospects for getting across the finish line are very much a reflection of collaboration. And this particular example illustrates that nicely, but more broadly between the patient community, the investigators, the funders. So I want to leave you with how valued and critical this team approach is, including everyone in this room's role. So, and then lastly, how that led to some of the biology that suggested that this urate target could be protective, and relatively quickly how we've been trying to move through therapeutic development. Right, so that's, I'll go through these things fairly briefly. So there's urate. Without dizzying you unduly about biochemistry, urate, you can see here, is the end product of a metabolic pathway that starts with something called purines, famous in our cells. Rock stars, if they paid attention to these sorts of things, adenosine being one of the key molecules of life in the genetic code and energy in our cells, but it still gets managed and it gets broken down to something called urate that largely in humans was considered an end product, a metabolic waste product, until these studies that and really the evolutionary biology that suggests that urate might be doing something very important in us. That's because in almost all mammals, urate gets broken down by an enzyme called urate oxidase to a metabolite, but not, as I mentioned, in humans here. It's us over there on this tree, our great ape brethren, and as well as lesser apes. None of us have this enzyme that breaks down urate. And therefore, we have fairly high levels of urate. If you look in the genetics of all us hominoids, there's mutations, actually multiple mutations, in the gene that breaks down urate, suggesting that something, a long time ago, you can kind of track it back to tens of millions of years ago, something good arose from having these high levels. And that led actually, it was theoretically proposed that since we lost our ability to make our own vitamin C a long time ago, that may have suggested that urate could serve as an antioxidant function. Bruce Amen colleagues demonstrated that urate is actually the main antioxidant that circulates in us long time ago now, and that led the epidemiologists who were trying to study these populations of people who are healthy, but a percentage of them go on to develop Parkinson's disease. What is it in their environment or in their blood that might be a predictor? And this is work from Alberto Escario at the School of Public Health here, this is just one example from a cohort called the Health Professional Follow-Up Study, where people who were, these were healthcare professionals, doctors who were followed for decades, and their blood samples taken earlier were just in them was measured this antioxidant, urate levels, and people had lower and higher levels, just a normal spread. And then following who developed Parkinson's disease, it became clear that people who had higher but still normal levels were less likely to develop the disease. And that observation, which has been substantiated by in many other cohorts, raised the question, if people who are healthy and have higher levels of this natural antioxidant, if they're less likely to get the disease, how about people who already have the disease? And there we turn to another collaboration of clinical colleagues in a group called the Parkinson Study Group in North America that's been doing these clinical trials, including many of those failed ones. to ask the question, if people who have the disease early on and have higher levels, does their disease progress at a slower rate? Here's a photo harkening back to the start of the organization over one of those failed trials. Vitamin E and Depronil were tested. You can see data top is on my colleague's shirts. David is an active member of the group too, and there are a number of trials that are ongoing and just starting up that we're excited about. But this is just one example, and this is what the answer that we got to that question of people who already have the disease. This is in two of those failed trials. One was called the DataTop, the one from the 1980s, and one called Precept. That was also listed on the tombstone part of the graveyard there. They didn't work, but we were able to go back through this sort of community approach and where these patients gave blood and actually spinal fluid and found this is their blood, their serum levels of urate, and looking at who progressed, got worse during the trial. And people who had higher levels were less likely to progress to the endpoint in the trial. In one of those trials, there was brain imaging that was done, as Jerry mentioned. We do something called a DAT scan. This is just an anatomical structure that early in disease, there's no easy way to see a difference between the brain of someone without the disease or with the disease. But with these DAT scans that some of you may have had, I agree, generally in a movement disorders practice of limited value if it isn't already obvious that someone has Parkinson's or Parkinsonism, degenerative versus essential tremor. It's of limited use, but in a research setting, it can be very useful because not only can you see differences in the amount of signal in that brain region, but you can also, where there's less of the markers of those dopaminergic nerve terminals with the DAT binding to them in someone who's developed early disease, usually with an asymmetry that reflects the one side having symptoms earlier. But you can also use this as a marker experimentally to track someone over the course of the disease as their scores on standardized tests for Parkinson's features get worse, that you can measure the signal that is being lost in serial exams. And that was done in one of these studies, and here this is everybody as opposed to men and women, and it's the same trend that people over the 2 years of the study on average lost some of the signal, and the amount that they lost, particularly amongst those who had higher levels of urate in their blood, was less over those couple of years, suggesting that there was a sort of biomarker of reduced progression in people who had higher levels of urate. So this was an unusual clue that drove this particular target that on the biomarker end, that higher levels could be a predictor of not only of a reduced risk of getting the disease, but also a reduced risk of its progression. Not absolute higher levels. You know, it's a fraction of a difference, but enough that was robust and enough to be a clue, especially when it converged with evidence of the biological plausibility that urate might not only be a marker of better outcomes in Parkinson's, but also could be mediating that. Obviously, it's an antioxidant, so that fits with theories of progression being oxidative damage driven. And then in the laboratory, many labs around the world have shown that elevating urate can be protective. I won't dive into examples. This is just illustrative from a group in Paris looking at dopamine-producing brain cells in a dish and seeing which ones survive, and very few do as you let them age. In this particular setting, that could be rescued or prevented with higher levels of urate in the dish. And similar studies have been done in animals, suggesting that really we had a good enough clue that was a little bit different than most of the previous ones. Still, no guarantee that this is a causal association, and ultimately the experiment that will give the answer is a human trial, a well-done trial, and that's what we were able to start about 10 years ago now, with the Fox Foundation supporting an earlier stage, mostly focused on safety trial. And I should say I'm pointing out urate as the candidate, but it turns out, kind of like dopamine being missing in Parkinson's, you can't just take, and you can't take that as you know, because it doesn't get across the blood-brain barrier. Similarly, taking urate doesn't work to elevate one's levels because it doesn't get across the gut barrier. But it turns out amongst ways to try to elevate urate, we had a good option in a precursor called inosine. That's urate. Inosine is not that much different. It was a few steps up in the pathway, and if you take inosine, one can elevate one's levels of urate. So that's what we began to do, and initially in a phase 2 study that was focused on safety of urate elevation in Parkinson's disease, or SURE-PD. Again, a team approach focused on safety because, as some of you may know, urate, which is also called uric acid, we evolved to have such high levels because of that presumptive benefit a long time ago, so high that it circulates close to the limits of solubility, and if it comes out of solution, you get crystals, and if that happens in a toe, that is particularly painful, and that can be as cartooned here in gout, also kidney stones if it happens in the urine. So we wanted to be very careful. We did a phase 2 study that was designed unusually based on the epidemiology data. There's that same slide, and with the idea in a sense of a precursor to precision medicine, where we just wanted to take the people who are at the greatest risk of progressing and who might be most safely treated with something that raises urate. So we just took people who had lower levels of urate and then randomized them to get placebo or one of 2 doses of inosine and then titrated to their blood levels of urate. This just shows, this study was published a few years ago now, that we met our endpoints that were mostly to show safety, which happily there was no gout and a few kidney stones, which we have to be very careful of. But we were very much able to elevate taking people who only had lower levels of urate and elevate them into these higher ranges and reversibly after 2 years, and found that, again, there was no gout, there were some stones, and we learned some things about how to best avoid them going forward, or at least try to. Although David is an investigator in our phase, he's not going to be talking about that, but I just bring up that no fault of his own, I know at least one of his patients, maybe 2, have had kidney stones, which is something that we're working very hard to avoid in the phase 3 that I'm just leading up to, but in telling you that we showed safety and tolerability, and also some evidence that people who are in the higher dosing of inosine into this moderate range of urate elevation had some evidence of slowed progression. Led us to a phase 3 trial now supported by NINDS, highlighting once again the collaborative nature, working with scientists, clinicians, statisticians that help you get things right. I often point out that they help me avoid mistakes. Even though reflecting my view of what we're trying to do, in that case, upping urate for safely in Parkinson's, or what I had suggested to be the Up Yours PD study, they help me avoid making mistakes. So it's a team effort. This is the phase 3 trial that's ongoing now. It started a year or 2 ago and will end in 2020. It's using inosine in a U.S. study that where we're hoping now to answer this question more fully, and doing the study in some ways that reflects advances on the clinical science. With again, we screen, we take advantage of knowing that people have lower urate, have faster rates of progression slightly, and bring only those into the study. And then we also are using imaging to as a screening test, not to make the diagnosis, but to help rule out people who don't have the disease, so that we can, with smaller numbers of subjects, treat them as shown here in this schematic for what a patient experiences after that screening process. Those with higher levels of urate don't come in, and those with lower levels do, and they get randomized to inosine to raise them, or stay on placebo at lower levels, and then after 2 years, we'll see where people are at. I just wanted to highlight that there's a lot of promise. The pace of the research, like Gia's group's doing, is supporting that from below. And not only are there some promising candidates in phase 3 and phase 2 that are coming up from environmental and biomarker clues, but there's a whole raft of promising candidates that we'll hear a little bit more about based on the genetics. And I had last couple slides just highlight the interaction of genes and environment and how exercise is a great outcome from that. But I've lost track of the time, and it's really ancillary. So if it's at, I think, let's see, 10:30. I think actually a time just to close out. It's really not much except because while we have all this excitement in saying, you know, the earliest studies are going to be here in 2 years, and the genetic clues are just getting into the clinic and might have a 5-10 year timeline to get their answers. What can we do now? And some of it's common sense. Some of it is informed by the science. And I think it's just always good to know that there's things that you can take charge of as well, in addition to volunteering for trials and giving blood samples, as many people in the audience I know are doing. So pesticides have, as an example of a risk factor that's been linked when good epidemiological studies like Alberto's from across the street have looked at people over decades and seen what predicts the disease. Just exposure to any pesticide, you know, not a huge effect, but stands out as a clue that fits with the toxins and their effects, like many pesticides have on oxidative protectants in our own cells, that can lead to not only some simple recommendations, but also as the science and the biomarker work has converged with the genetics. You'll be hearing about synuclein a little bit more, but there's evidence that these pesticide and oxidative pathways interact with genetic clues, and the alpha-synuclein being one. So take-home message, continue to enjoy gardening, just use gloves and common sense and the like. And then lastly, there are a lot of environmental exposures that have been linked slightly or less, and most of them are in the margins of being able to offer dietary or exposure advice. One, despite not, one exposure or lifestyle feature that almost everybody knows at some level instinctively and have heard recommendations for, just to tie it a little bit to the to some of the biology. Actually, Alberto's group and others have also highlighted people who exercise more actually are less likely to get the disease. And there's some biology, just as an example here, linking synuclein levels with exercise. Too much synuclein seems to be a bad thing, and people who have lower levels, that correlates with exercise. How that interaction takes place is something we're sorting out. But the take home here, simple enough, is something after sitting here and hopefully taking a lot in, activating your minds, activating your bodies on a regular basis, you wouldn't go wrong. So anyway, I wanted to leave you with that, as well as illustrating the example of the hopefulness through some of the neuroprotective ideas that are in the pipeline and getting close to the finish line. And then lastly, the collaborative theme with everybody in the room represented here on it. So thank you.
So I would just take a few quick questions before the break here. Actually, I'm going to take a minute to look through these. One question is, do you want to say anything about people who wonder if they should take urate? Or take inosine, I mean?
Sure, thanks. That's always good in balancing the enthusiasm, particularly with some of these exposures that are readily available. Inosine is a nutritional supplement that's available over the counter, but it's I and the Fox Foundation, others, don't recommend trying to elevate one's own urate levels, in part because of the kidney stones and the gout potential. We know we have to do a lot of work to very closely fine-tune to avoid kidney stones, and given that it is promising but unknown, the balance of risk and benefit doesn't warrant that at this point.
Another question, are there things people can do naturally, like diet, to change their urate levels?
They can, but again, the idea is that pushing that it's hard to do actually dietarily. To repeat the question? Yes, thanks. It was just whether one can do anything dietarily to increase urate levels. I didn't show, but urate levels are determined by, like many things, a mixture of your genes and environment. Environment in this case is what you eat. About a third of one's levels are determined by one's diet. And in fact, some of the epidemiology studies have shown not just looking at blood levels of urate being linked to not getting the disease, but people who have diets that produce higher levels of urate are at a slightly lower risk. It's actually very hard to do to eat so much purines, these precursors that get broken down into urate. And if one does that, one might be at increased risk for these stones, and more likely, if one just does that, one's not going to make enough of a difference to actually get into those zones of better outcomes. So at this point, I think it's more a source of hope than a dietary guide.
And you mentioned similarities with vitamin C, an antioxidant. Should people take vitamin C?
Yeah, so there haven't been as advanced assessments of other natural antioxidants, including vitamin C. Vitamin E was actually the number one first candidate to be laid to rest back in the 80s. And so so there is no clear evidence that, despite some of the associations and the theory that antioxidants will help. And I should point out that vitamin C and vitamin E, there's no data, unlike the urate, or there's some other links, there's no epidemiology that suggests people who have higher levels of vitamin C are more likely to avoid the disease.
And maybe even though there's a lot more questions, I'm sorry we don't have time for all of them, maybe we should stop there and give people a moment to take a break. So thank you very much.
Thanks.
Maybe 10 to 15 minutes to start again.